Hello everyone, my name is Hyunjin Chang from the Hyperlab team, and in this video we'll be showing you how we could use Pirtobrutinib drug to rediscover hit candidates with having the similar characteristics. To begin, Pirtobrutinib is a BTK inhibitor which is non-covalent, and it can both inhibit the wild type and the C481S mutant. And because of this, it's been approved by the FDA in 2023. So in Hyperlab, in order to work on your drug discovery, you must first register the 3D information of your target protein, because Hyperlab is based on structure-based drug discovery methods. So if you head into the project and click the protein structure tab, it's going to show you the page with the protein structures. And usually if it's the first time coming into your project's page, it's going to be empty, so you'll have to add or register a new protein structure. Usually you can search your target protein by the given PDB ID. If there is no crystallized structure in the PDB, you can also use a predicted structure from the AlphaFold. Or if you have a modified something that you made manually from other softwares, you can also upload your target protein structures in PDB format. So if we search for the ID here, it's going to show you the protein structure and the ligand bound to it. So this is actually AFLL is the BTK with Pirtobrutinib bound to the binding site, and AFLN is also Pirtobrutinib bound to the C481S mutant BTK. From here, you can set the binding sites based on the position of the ligands inside the PDB. So if you want to, usually it's set to the ligand from the given ID, but usually if you want to change the binding site, you can always set it to something else. Or you can also create a binding site based on the position of the binding residues, or if there are no ligand bound form of your target protein. So for instance, if we choose a certain residue, it's going to be showing you the binding site will be set on a very small scale, because this box will be given by the size of the residue or the ligand that is of your selection. So if you're to do so in this instance, you have to actually change the center of the box, or increase the size of the box in a sufficient manner. Usually 20 angstroms is good enough. So and if we can increase this, then it's going to be sufficient for you to work on your bind docking process or virtual screening. However, in this case, since we already have a ligand bound to the receptor, we're going to be using this site as our to work with. Once you're done with setting the binding sites, you can just click save, and that will be registered to your protein list. Once you do so, you have to activate the protein structure by clicking the inactive or active button. And once that's done, you're ready to head to the bench page. Currently, we have two bench pages. One is My Bench, and the second one is Project Bench. The difference is between My Bench and the Project Bench is that if you add a molecule, or if you put anything inside this My Bench page, the members inside this project will not be able to see your work. But if you put in a molecule in the project bench, you'll be sharing this information with the others. So please note that. So currently, we already have some results. But first, what we did was we actually added the reference molecule, Pirtobrutinib, to check how much it's going to be in alignment with the actual experimental tendencies. So here we have two binding scores, the binding score from the wild type and the binding score from the mutant. And from here, you can see that the binding score is minus 11.4 and minus 11.3, respectively. And this coincides with the experimental tendency that pirtobrutinib can both inhibit the wild type and the mutant structures. Usually, if a binding score is around bigger than 7 or 8, we usually consider the molecule to be having a pretty high binding affinity. But this is not the absolute value. It usually differs by the actual experimental data or the size of the molecules or the size of the binding sites. But when it's usually bigger than -9 or minus 8, we usually consider the ligand to be having a pretty high binding affinity. And also, if we can further investigate the results from the docking results, which is hyperbinding, by seeing the 3D pose of the docking results. So we have the 3D viewer that's from the molecule list, and if we just first check the X-ray structures, the pose of pirtobrutinib from the X-ray crystal structures, and compare this with the actual docking results, you can see that there are some few differences between the X-ray structures, but the core backbone is actually in alignment with the X-ray structures. So we can say it's pretty in a good alignment with the actual experiments. You can also check the other conformer poses. These conformers are actually lined up based on the binding predictions. So you can also look for other binding poses. We usually give a maximum of 10 conformers, but if there are poses which there were predicted less than that, it's going to be showing you less. You can also check the 2D diagram between the ligand and the target protein interactions using the 2D diagrams. So from here, you can see that with the conformer with the highest binding score has a hydrogen bond with methionine 477 with a hydrogen bond distance, which is shown here to be 2.85 omegrams. You can also check some other features such as the binding pocket conformation by increasing the surface opacity. And from here, you can check how the binding pocket is formed and also how the given ligand is actually posed inside this pocket. And since we have confirmed that the reference ligand, pirtobrutinib, is actually showing a similar tendency with the experimental values, we can now start screening to look for a new scaffold or hit candidate. Currently, in hyperscreening, we provide 5 built-in libraries, fragments having 500,000 small molecules, having a molecular weight less than 300 kilodaltons, while p-value is smaller than 3, and hydrogen bond donors less than 3. And these are for fragment-based drug discoveries, so keep that in mind. We also have the kinase library, which has 65,000 molecules specialized for kinase interactions. These usually are made up of pin binding motifs. We also have some allosteric kinase inhibitors inside this library. We also have 1,100 FDA-approved drugs and also 4,200 molecules made up of natural product-like fragments. Lastly, we have the diverse library, which has 1 million molecules having diverse structures and diverse properties. This is usually most fit for discovering a new scaffold or candidates for your given target protein. And this, if you select this, it's going to be showing you how much screening point it's going to use. And once you start screening, it's going to actually take around 24 hours, give or take. Currently, we already have the results from the screening, and here we use the kinase library to screen for a new hit candidate. And from here, we sorted the molecule list based on the binding score. And from here, we start to rule out molecules based on the binding score first. So if you click the filter button, you can filter this molecule list based on the property you wish to filter out. So for here, if we want to filter out a value to be less than minus 9.4 kilocalories per mole, binding prediction values greater than 9.4. The reason we picked 9.4 is because the value is the binding score value of Pirtobrutinib is minus 11.4, minus 11.3, and we wanted to rule out molecules having binding scores having a difference more than 2 kilocalories per mole. And the second characteristics we want to apply the filter on is the binding score difference between the wild type and the mutant. As I have told you, Pirtobrutinib can both inhibit wild type and the mutant. And if we have and these were also shown with the binding score values. And if our candidates have a binding score difference between the wild type and the mutant, it would not mean that the given candidate or the molecule will be able to both inhibit the wild type and the mutant. So in this case, the first molecule, which has a binding score of minus 8.9 kilocalories per mole and minus 10.6 kilocalories per mole for wild type and the mutant respectively, is not suitable for our candidate. And the next one, which has minus 10.8 and minus 10.6, this has a small difference, so this will be suitable. This seems like a suitable molecule to work on. And from here, we can also check the results from the ADMET prediction values by clicking the result icon. Once you do so, it's going to show you the detailed information of your given molecule, the molecule name and the SMILES code, and also the purchase link of the molecule. This purchase link is only provided to molecules from the hyperscreening molecules. So once you click this, it's going to lead you to a page where you can purchase this molecule. And on the bottom, we also have the physical chemical properties of a molecule, which are calculated. And on the right column, we have the ADMET prediction values. So currently we provide the log P value and the solubility, metabolic stability, CYP inhibitor, CYP substrates, hERG, PGP, PAMPA, drug likeness, synthesis complexity, and BBB. The synthesis complexity gives out a certain number which requires you to synthesize this molecule. And usually if this is greater, it means it's much more difficult for you to synthesize. And the drug likeness value is a score for to see if this molecule is similar to a structure to other known drugs. So as you can see here, FDA-approved drugs have a drug likeness score of 74.5, and Kemble has 60.5, and GDB17 molecules have 40.5. And since our molecule has a score of 62.9, we could say it's pretty suitable. It has a bit of a drug-like structure. You can also check if this molecule is novel or not in an indirect way by searching for other similar compounds. And usually you can we have actually calculated or actually searched for the similar compounds already, but if you did not calculate this, there will be a button to search for other compounds which are similar to the given molecule. And once you do so, it's going to give out molecules which have which has a similar structure to your given molecule by the structure similarity. by default, the similarity threshold is given as to be 90%. And the molecule on the top left is the one which has the highest similarity score. And the score decreases as you go on. And since the first one doesn't have a lot of patents, or it doesn't have a literature, it only has a single patent, and the one next to it also has a single patent. So we can say that the molecule we see here is actually pretty novel enough, so to speak. However, so in order to, we can't set this as our hit candidate, but we also we can improve the properties of this molecule by using the molecule designs. So if we look at the ADMET properties, we can see that the log p-value is 4.6 and the solubility is moderate, but the metabolic stability is given out to be very stable. So in order to improve the metabolic stability, we can perform molecule designs to improve those properties, and that is actually shown in here on the details page. Or also, you can also head to the design page by clicking the continue or start button from the Hyperlab column. And once you click this, it's going to lead you to this design page. And if you click the plus button on the molecule, it's going to show you the another page where you can actually edit the molecule structure from the molecule editor, or you can also use the hyper design features, which can actually make you replace or add certain functional group in this molecule. So if we click replace, you can select the functional groups that you wish to replace and start run, and it's going to give out other derivatives replacing this functional group. If you want to add another certain functional group, you can click add, and then it's going to highlight all the valence atoms where you can actually add these functional groups to. You can select one and start running, and it's going to show you derivatives with new functional groups added to this atom. Currently, we have a design calculation log. We already performed this previously. And once the calculation is done, it's going to show you this results page made up of 50 derivatives having based on the highest binding prediction values. From here, you can check the binding scores, log P, TPSA, and molecular weights. And since we wanted to improve the metabolic stability of the given ligand, we can actually look for a molecule which has a lower log P value, because lower log P would indicate a more improvement in metabolic stability values. And so if there are other type of log P values which are smaller, and with something that would also increase the binding score, this third one is actually the only one having suitable conditions. So we actually added this already. And once we compare this with the some of the 3D pose, we can kind of check how the molecule new derivative has changed. We can also change the color of a molecule from this column. And as you can see, the binding pocket, with respect to the binding pocket view, we can see here that the functional bit that we replaced here, it has been changed to a different functional group. And we can also check if there has been any difference in the interactions. So this is the actual, this is the 2D diagram of your derivatives. And from here, you can also check what kind of interactions has been added, or what sort of differences have been made, not just by the 3D pose, but also from the 2D. So this is sort of the main features of the Hyperlab platform. Once you are done with this, you can also download your data by selecting the molecules and downloading these information in a CSV format or in an SDF format. The SDF format has also the 3D information of the ligands, so please do note. And thank you for watching this video, and I hope you can achieve some impressive results using HertoGuide. Thank you.